This first talk that I'm going to give is called Observational Studies. What are the problems with observation? It's sometimes hard to figure out what caused what. And as we'll see, there's something called regression to the mean that could explain what you're observing. There's the concept of association doesn't necessarily mean causation. And then there's the problem of confounding and bias. And I'm going to give you three examples, three interesting examples, one on falling cats, one on lawyers in cooperating with police, and I'm going to add a personal touch on that one. And the third is when to start antiretroviral therapy for people with HIV. I'm going to talk a little bit about the common types of observational studies, cross-sectional, case control, and cohort. And then I'm going to talk about a so-called self-controlled design that I think has some advantages. And I'm going to illustrate that with a study that we did on the H1N1 vaccine and Guillain-Barré syndrome. And then I'm going to tell you about an interesting controversy involving hormone replacement therapy. So observation is the first step to discovery, and here's some famous cases of observation leading to discovery: hand washing and reducing infection. Edward Jenner noticed that milkmaids didn't get smallpox, and that led Jenner to discover the smallpox vaccine in 1796. Cases of cholera in London tended to cluster around a water source, and that led to John Snow discovering that the foul air theory that was present about what caused cholera was discounted after that. So some famous examples of observation leading to discovery. Now, what's the problem? It's easy to misinterpret observational data, and I'm going to give you an obvious example, which is cholesterol medications. And let's suppose you want to know the effect of cholesterol medications on cholesterol. Well, if you just foolishly said, I'm going to compare everyone on cholesterol medications to everyone not on cholesterol, to people not on cholesterol medications, then of course the people who are on the cholesterol medications have higher cholesterol, sorry. But that doesn't mean that the cholesterol medications cause the higher cholesterol. That means that the people with highest cholesterol were the ones who were put on the cholesterol medications. So it was the high cholesterol that caused them to be put on medications. It wasn't the medications that caused the high cholesterol. And so, most people would realize there's a problem with that. And so you could say, well, let's look at a different way to do it. Let's look at people who have high cholesterol and look and see what kind of change in cholesterol you see after they started their medications. But suppose that their cholesterol does decrease once they start medications. Can you conclude that it was because of medications? It could be that that there were other changes at the same time. Maybe the person started exercising more and eating a better diet because their doctor told them they had high cholesterol. Or maybe the reduction in cholesterol was due to something called regression to the mean. Now, what is that? What is regression to the mean? Regression to the mean says that people with extreme values of something, cholesterol, blood pressure, whatever, tend to have less extreme values if you measure them again later, even if you don't treat them at all. And part of the reason, the reason for this is that the reason the original value was extreme, part of that reason is measurement error. And so given that you had a high reading, the amount of error is actually likely to be greater, and it's likely to be positive. In other words, the error in reading cholesterol tends to give a high reading if the person had such a high reading. And so the next time you measure them, the measurement error is going to be, centered around 0. And so when you look at them again, if they were high on the first reading, they'll tend to be lower on the second reading. If they were low on the first reading, they'll tend to be higher on the second reading. And I'm going to give you kind of a pretty cool example, I think, of regression to the mean in action. This is in a big study of about 1,700 people, and there was it was a randomized trial. And to get into this kidney sub-study of the trial, you had to have estimated glomerular filtration rate less than 60. That's a kidney measure. And so low values are bad. And so they identified a group of people with EGFR less than 60. And then here are the EGFRs over time in that study. And these are the people who had less than 60 at the beginning of the study, times 0. And you see they jumped way up after baseline. The first month they went way up, and their EGFR stayed much higher after that. And so people were concerned in this study, and they said, well, it must be that people were dehydrated at baseline, and then they got better care in the trial, and therefore they did a lot better. But actually, a big reason for what you're seeing here is regression to the mean. If you take people who had to have very low values to get into the study, when you measure them again, they're going to have higher values even without any treatment whatsoever. And to show this, what we did was we said, okay, instead of identifying those with EGFR less than 60 at baseline, we're going to look at the 1 month value, and we're going to identify a group of people whose value at 1 month is less than 60. And then what we're going to do is we're going to go backwards in time. We're going to say, okay, for that same group of people, what was their EGFR at the beginning of the trial? And what you see is a somewhat similar picture. And so when we do that, here's the group that we identified that had low EGFR at month 1. And now you see that if you go backwards in time, their EGFR is higher, meaning they're doing better, their health is better. And so this discounts the theory that, oh, it was just that they were dehydrated at baseline, and then they came in and got better care, and that's why they had better values. This, again, this is identifying a group based on their 1 month value, taking that same group and looking backwards, and you see they're doing better. So what this is really saying is anytime you identify a group of people who have very low values on some measure, and then you measure them again, you're going to see something that regresses toward the mean, toward the more normal values. And this is a very interesting phenomenon that happens. So part of the reason you're seeing values go lower when you give the cholesterol medication, part of the reason for that is regression to the mean. Now, certainly these medications do lower cholesterol, but you do have to account for this regression to the mean. All right. Okay. So let's return to that study of the effect of cholesterol medications on cholesterol. So an alternative design would be to look at 2 groups with high cholesterol, one group that chose to go on medications, and another group that chose not to go on cholesterol medications. And and then seeing what changes the 2 groups had in terms of cholesterol, and seeing if there's more of a change in cholesterol in the group that went on medications. But there's a problem with that too, and that is that the people who choose to go on cholesterol medications might be the kind of people who are just more health conscious in general. Maybe they get more exercise, maybe they eat a healthier diet, they might go to the doctor more often, or they might just have a more positive attitude and thinking about their health. And all of these things, including having a positive attitude, I believe can have an effect on your cholesterol. Now, it's hard to adjust or, even measure some of these things like, the positive attitude. How would you measure that? Um, and so you can never really be sure whether the difference that you're seeing is because of the cholesterol medication or because of some features of the participant that make them more likely to take cholesterol medication, including these other health conscious behaviors. So the solution to this problem is to randomize patients to the two different arms, randomly pick patients to get the cholesterol medication and then to take no medication or placebo. Um, and then if the trial is blinded, in other words, they don't know whether they're getting a placebo or they're getting the active medication, then the two arms are going to be balanced with respect to the percentage of people who get more exercise and who eat healthier, and even the percentage of people who have a positive attitude should be very similar because a coin flip is deciding which group they get into. So there shouldn't be any systematic differences. Now, so that's the solution, is to do a randomized controlled trial, which we're going to talk about later. Um, but another thing that you have to be aware of is that there can be a association but not causation. So two factors, A and B, for example, A might be taking the cholesterol medications and B might be having high cholesterol, can be associated without A causing B. And in that example that we talked about, where we compared people taking cholesterol medication and not taking cholesterol medication, if we just look at those two groups of people, we saw that no, it was actually B causing A. It was the fact that people had um, high cholesterol that caused them to take the medication. It wasn't the medication that caused the high cholesterol. So it might be that B causes A, and you thought that A caused B. In other cases, something else could cause both A and B. So, for example, there is an association, you'll see this, there is an association between carrying matches and having lung cancer. And of course, the reason that there's an association between those two things is that smoking is what causes people to carry matches, and it also causes lung cancer. So there's a third thing that's causing the A and B, and smoking is causing them to carry matches and to have lung cancer. So it's crucial to look at biologic plausibility. There's no biologic reason that carrying matches should cause lung cancer. It's much more plausible that smoking causes lung cancer. You're introducing toxins directly into the lungs. And some of the other factors to consider when you're trying to assess whether there's a cause and effect relationship. First of all, is there a statistically significant difference? What is the strength of the association? Is there a dose response relationship? Do you see that people who smoke more often have higher rates of cancer than people who smoke less often? Is there a temporal association? Did the disease happen relatively soon after starting the risk factor? Is there biological plausibility, as we discussed? And are results replicated in different studies? Now, one thing that's well recognized in observational studies is if you see A and B occurring together, it's hard to figure out which one necessarily causes C, if either one of them causes C. And this is called confounding. And so a good example of this is that alcohol abusers often abuse other substances as well. And so if you're trying to look at what is the association between drinking during pregnancy and birth defects in the baby, it's going to be confounded by other drug use during pregnancy. So you won't really know whether the drinking caused the uh, birth defects or the other drugs cause the birth defects. And so what do you do about that? One common thing that you can do is called stratification, where you look at the association between, in this case, the amount of drinking and birth defects separately within people who abuse drugs, other drugs, and within people who don't abuse other drugs. Of course, you have to, you'll never know for sure whether they abuse other drugs or not. But that's one way to try and look at things. Or you can also use a statistical model to adjust for the fact that people are abusing other drugs. And so what you would do in that case is in your statistical model, you would include the amount of drinking, and then you would include a term for other drugs. This approach is not quite as easy to explain. And there's the problem that you may have the wrong statistical model. But those are two ways that people often adjust for other factors. There are worries in observational studies about comparability of the groups. At baseline, are these two groups similar with respect to other factors other than the thing that you're studying? And did they receive, during the period you were following them, did they just receive differential background treatment that could also cause you to misinterpret what's going on? There are also different biases in observational studies. You may detect more events in one group than another because maybe there's more frequent monitoring. The doctor might be looking more closely at them if the doctor believes that they're engaging in some activity that's risky. And maybe you're more likely to suspect that there was a certain event if you know that the person is in a certain risk factor, has a certain risk factor. You might say, okay, I think my suspicion is confirmed that they did have this this event, this heart attack, or whatever the event was. Then there's also the problem of stimulated reporting of adverse events. This is called a reporting bias. Once you hear that some drug causes kidney problems, then everyone who has any kidney problems is going to say, yes, I did take that drug once, didn't I? And so you're going to have more reporting of these adverse events. And there are often biases in the way data are collected. Now, I'm not saying that observational studies, can't be done well. They certainly can, but these are things that you have to worry about. And my first example, I borrowed this from Annika Grobler from South Africa, Durban. And this is about cats. There's a New York Times article that's called "On Landing Like a Cat, It Is a Fact." This is in 1989 published. And these two veterinarians, Wayne Whitney and Cheryl Melhaff, recorded the distances that cats fell, 129 cat falls, and they noticed that the mortality did not seem to increase with the height of the fall. And of course, that didn't make sense. You would think that, if they fell from a higher distance, they're more likely to die. So the problem, though, is that dead cats are not taken to the veterinarian. So these are veterinarians. They're looking at cats that were brought to them. Obviously, no one is going to bring a dead cat to them. because there's nothing they would be able to do for them. So only the cats that survived the initial fall are going to be brought to them. So there's a bias because you're not seeing the cats that died and were not brought to the veterinarian. So that's an interesting bias. When you think about it, you realize, oh yeah, that's a problem. Now, this next example is a little bit controversial because I'm going to bring a personal touch to this. This is me in about 1977. And so let me just give you some background. So I had had an argument with my girlfriend. I thought it was relatively minor, but apparently she didn't. And next thing you know, the police were asking to search my car, and because they said they had been told that I had marijuana in the car. So I agreed to the search, and lo and behold, there was marijuana in the car, and I got arrested. And I talked to my lawyer, and my lawyer said, "Oh, you made a big mistake. You should never have consented to the search. You should never cooperate with police." And this is a common advice that lawyers give, not to cooperate with the police. And their evidence, what is their evidence that you should not cooperate with police? Their evidence is that their clients cooperated and got arrested, right? They are only seeing the people who got arrested. There may be many other people who cooperated and did not get arrested because of the fact that they cooperated. They allowed the search, and maybe the police felt sorry for them or whatever, and they like the fact that they cooperated and they don't arrest them. So that's a little bit of a bias, and so that makes it difficult. So lawyers are not seeing the full evidence. Next example is when to start antiretroviral therapy. This is for people who have HIV, and HIV kills CD4 T cells, these so-called helper T cells. And so there was an open question about when you should start antiretroviral therapy. So starting when you have a very high CD4 T cell count preserves immune function, but there was a thinking that perhaps some of these medications cause some cardiovascular or renal events. So there was an open debate about whether you should wait for the CD4 count to go down to a relatively low level, or whether you should start antiretroviral therapy, early in the infection. So let's suppose that you were trying to do an observational study to compare whether it's better to start antiretroviral therapy when your CD4 count is 500 versus allowing your CD4 count to go to 350. And if you just look at everyone who started therapy at 500 versus people who started at 350, there's an obvious bias, because why are doctors starting some people early? Why are some people starting them at 500 instead of 350? And they're doing that because perhaps they had some serious infection that they thought needed to be treated, and so they're starting people at 500. They're starting sicker people at 500 versus the people they start at 350. Now, there's more subtle biases that can also come into this, and one of them is very interesting. It's called lead time bias. So suppose you're looking at people who started at 350, and you're looking at people who started at 500, different people. Then if you're looking at those people and you're saying, okay, how long does it take from when I started observing them till they get an AIDS-defining infection, AIDS-defining event? And So when the question is, when do you start the clock? So if you say, I'm going to look at the time from when they started the meds to when they got an AIDS defining event, then it's clear, even if you took two people who would have had AIDS at exactly the same time, if you look at the one person who started at 350 and the other one who started at 500, this time from when they started to when they get AIDS defining events is going to be shorter for someone who started at 350. So that's going to introduce a bias. If you start at the time that they reach 350 versus the time they reach 500, then it's clear that the time from 350 to the AIDS event is going to be less than time from 500 to the AIDS event. Now, but so the other thing you could do is you could start the clock at when they had CD4 T cell count of 500. So for this person and this other person, you're starting the clock when they had a CD4 count of 500, and you're saying, okay, I'm going to look at the time from reaching 500 to when you had an AIDS event, and I'm going to look at that for the people who started therapy at 350 versus the people who started therapy at 500. The problem with that is for these people who started at 350 to get into your study, for these people who started at 350 to get into your study, you have to know that they didn't die, for example, in this period between 500 and 350. And so these people, they did not die during this period, whereas these people who started at 500 might have died during this period between 500 and reaching 350. So that there are going to be no events in the 350 group between 500 and 350, and yet there might be some in this other group that's started at 500. So that's another bias. How do you deal with that? There's all kinds of problems that don't occur in a randomized controlled trial. And so because of the potential for this bias, the National Institute of Allergy and Infectious Diseases, which is where I'm from, sponsored the Strategic Timing of Antiretroviral Therapy, or Antiretroviral Treatment, START trial. It was a randomized controlled trial. They took patients who had HIV and had CD4 counts that are 500 or greater, and they randomized people to either start antiretroviral therapy immediately or wait until their CD4 count dropped below 350. And so this does not have any bias because you're going to count everyone, whether they had, there's no ambiguity about which group they're in because they're randomized to be in the 350 or the 500 group. Or I should say the immediate ART group versus the 350 group. And this trial was overwhelmingly significant. It showed that starting antiretroviral treatment immediately reduces both AIDS events and serious non-AIDS events. Now, what are the common observational studies? They're cross-sectional designs where at a single time point you ask people, whether they have a disease, let's say it's lung cancer, and whether they engage in a risk factor, for example, smoking. So you just find out for each person, do you have lung cancer? Do you smoke? And then you just see if there's an association between those two. Then there's also a case control study, which is a retrospective study where you take people who have the disease of interest, in this case, lung cancer, and then you study a group of controls. So this is a group who do not have lung cancer, and you see whether smoking is more likely among the cases, among the people with the lung cancer. And then same thing, you can also do it the other way around, a cohort study, which is retrospective or prospective, but the stronger types of designs are prospective studies So now instead of looking at cases and controls, you're looking at a group of people with the risk factor, in this case, smoking, and another group that don't have the risk factor, they're non-smokers, and you look forward in time and you see how many people get lung cancer in each group within a specified time. Now, cross-sectional studies are really cheap and easy. They're short duration. You're just going to, at a single time, find out whether they smoke and whether they have lung cancer. So you could measure a whole lot of other factors. You could, in your questionnaire, you could ask them about other conditions. But one of the cons is that you don't know which one came first. You just know at a single point in time whether they're smokers and whether they have lung cancer. You don't really know which one came first necessarily. And there are a lot of biases as well, and this holds for any of these observational studies. Now, the case control study, this is a really good design if you're studying a rare disease. Because if you're studying a rare disease, if you just follow people with and without a risk factor, you are not going to see very many people who get the disease. So you won't be able to tell whether that risk factor is causing disease. And it's not as time consuming and expensive generally than a prospective study looking forward. You can study multiple risk factors simultaneously, but one of the cons of case control studies is you really can't estimate the prevalence or incidence of disease. So with a prospective study, like a cohort study, you can say, okay, over a 1 year period, this proportion of people who got lung cancer. With a case control study, you really can't do that because you're studying a fixed number of people who got lung cancer and a fixed number of people who didn't. You really can't tell what the incidence of disease is or prevalence. And then there are all kinds of biases. You find recall bias. The people who do have the lung cancer are much more likely to remember that they smoke, and more likely to remember other risk factors. There can be measurement bias, interview bias, depending on how you ask the question, you get a different result. Selection bias, that is, there might be differences other than what you're looking at, your risk factor. You might be, when you look at smokers, when you look at people who have lung cancer versus people who don't, how do you know that they're comparable with respect to everything except smoking status? It's pretty hard. This is also not a good design if the exposure, the risk factor is rare. It's great if the disease itself is rare, but not if the risk factor is rare. Cohort studies going forward are better. You can see the temporal relationship between when you were exposed to the risk factor and when you had the outcome. And you tend to get less biases between the group that have the risk factor and don't have the risk factor versus a group of people with the disease and without the disease. You get natural history information. You can estimate incidence rates and risks, and there's generally less bias than in the case control study. But they're long, they tend to be longer, and they could be expensive to follow up all these people. You usually need large sample sizes and baseline data. And again, if you have a rare disease, you're not going to get much out of a cohort study because almost no one's going to get the disease. And as with all of these observational studies, you are going to encounter bias. Now, I wanted to give this example of what I think is, a pretty well-designed observational study. Of course, I think this because I was involved in this study. This involved the H1N1 vaccine, swine flu vaccination. And the question was, does this increase the risk of Guillain-Barré syndrome, which is a very rare neurological condition? There was an association back in 1976 between the H1N1 vaccine and Guillain-Barré syndrome. And so they wanted to study whether there was an association between this vaccine and Guillain-Barré syndrome during the 2009-2010 vaccination, because they had some evidence that suggested there might be an association. So they thought about different alternative designs. Maybe we should compare vaccinated people to unvaccinated people. But of course, people who get the vaccine are different than people who don't get the vaccine in many ways, perhaps. And so maybe it would be better, and I think it would be better, to compare the people who received the H1N1 vaccine to the people who received the ordinary flu vaccine. And those are more likely to be comparable in terms of how health conscious the people are. There's a little bit of a problem in that there might be a great overlap. Maybe most people who got the flu vaccine also got the H1N1 vaccine, so it might be hard, but to find enough people who only got one. But at any rate, there's another design that I think is a good one, which is the self-controlled design. And this is what we did. We said, we're going to look at people who had H1N1 vaccine and got Guillain-Barré syndrome within 91 days. And then what we said was, okay, if it really was the vaccination that caused the Guillain-Barré syndrome, we would expect to see a temporal relationship. So when we look at the people who got Guillain-Barré syndrome within the first 42 days, and then we compare those to compare that proportion to the proportion who got Guillain-Barré syndrome between days 50 and 91 after the vaccination, and we eliminated the period between 43 and 49 days, there should be, we should not see any difference, right? If the vaccination did not cause the Guillain-Barré, we should not see a temporal association. Remember, we're looking at everyone who did get Guillain-Barré syndrome, and we just want to know, was it within the first 42 days after getting the vaccination or not? And so we've got this little dot plot here. So this is days 1 to 42 after getting the shot, and this is days 50 to 91. This is the same period of time, and you can see that there are quite a few more people who got Guillain-Barré syndrome within the first 42 days. And so that suggests that maybe it is the vaccine that's causing the people to get Guillain-Barré syndrome. Now, and the reasoning is, if it didn't increase the risk of Guillain-Barré syndrome, then why should the proportion with Guillain-Barré within the first 42 days be so much higher than within the last 42 days? Now, there are other explanations that you could think of. There's confounding with regular flu vaccine. So as I said, people who get the H1N1 vaccine may also be getting the ordinary flu vaccine. So maybe it's the ordinary flu vaccine that's causing an increased risk of Guillain-Barré. Or maybe it's the flu itself. Maybe people are getting the flu, and that is increasing the risk of also getting Guillain-Barré syndrome. I think that is a fact, that flu does increase the risk of getting Guillain-Barré syndrome. And so maybe that's what it is. You're thinking it's the vaccine, but it's really the fact that they got the flu, and they got the flu, during the flu season, which is when people usually get the Guillain-Barré, get the H1N1 shot. And so maybe that's what's causing it. So what we did there is we said, all right, let's look within these different strata. So overall, what we saw is there were 54 episodes of Guillain-Barré within the first 42 days, versus 23 episodes, 23 cases of Guillain-Barré during the last 42 days. And so this is like flipping 77 coins, basically, and noticing that 54 out of the 77 are heads. And so when you look at that and say, how unusual is that? That p-value is less than 0.0001. So it's extremely unlikely. If you flip a coin 77 times, 54 flips, 54 or more of them would be heads. And similarly, this is striking evidence that there were a lot more Guillain-Barré episodes during the first 42 days after the vaccination. Now, to address the what maybe it's the flu vaccine that's causing this, we looked at the people who got no flu vaccine. So these are the people who got no flu vaccine, and you can see there were 14 such people, and 12 of them, 12 out of 14, got the Guillain-Barré within 42 days after the vaccination. So that suggests even in people who didn't get the flu vaccine, there's a striking difference in the time course of the Guillain-Barré. And then similarly, these are people who never got flu, or at least they never had flu-like symptoms. 42 events happened in the first 42 days, whereas only 15 events happened within the last 42 days. So even when you look within these various subgroups, it does look like there's a striking difference in the proportion of people who are getting the Guillain-Barré syndrome within the first 42 versus the last 42 days. And so this is highly suggestive. Now, the other thing that people said was, oh, wait, but maybe there's other kind of seasonal confounding. And so maybe it's just that they're getting the vaccine in certain season, the flu season, and so maybe there are other things going on at that same time. So what we did was we stratified by when they got their vaccination, and we looked at different months to see if there was a difference over these different time periods, in terms of the higher proportion in the first 42 days versus the last 42 days. That does look kind of interesting that during this time period, it's only 11 versus 8, which is not that unusual. But in all these other time periods, it is pretty unusual, the number of ones in the first period. So when we look at these things, by these different subgroups, we see similar results. And so that leads us to believe that that this is a real finding. There is an increased risk, or at least there was with this vaccination that they were using in 2009, 2010. Last thing I wanted to tell you about was a really interesting story about hormone replacement therapy. So the first hormone replacement therapy in postmenopausal women, or going through menopause, actually, it was approved by the FDA in the 1940s. There was people noticed that the cardiovascular heart disease risk in women was low until after menopause, and very low compared to men's risk. And then after menopause, the risk went way up. And so hormone replacement therapy was also believed to increase bone density and reduce the risk of fractures, and the evidence is strong in that area. So there was a large rise in the use of hormone replacement therapy in the 1960s, despite the fact that there were no randomized clinical trials. And then there was some issues over hormones causing cancer, and in 1975 there were two New England Journal of Medicine articles linking estrogen use to endometrial cancer. But then there were some new studies that showed if you just added progestin, that took away the risk of cancer. So there were about 30 epidemiologic studies that established the benefits of hormone replacement therapy, many, on cardiovascular risk. Then there was a big study in 1985 called the Nurses' Health Study that showed that women taking estrogen had 67% fewer heart attacks than women who never took estrogen. This was a large study had good follow-up, and many people were convinced. And then along came the HERS study, the Heart and Estrogen Progestin Replacement Study. That was the first placebo-controlled, randomized controlled trial for hormone replacement therapy. Had about 2,760 patients. And they found that hormone replacement therapy did not prevent cardiovascular coronary heart disease, is what CHD stands for. And they, in fact, they saw an early increased risk of coronary heart disease and an elevated risk of stroke and clots. So this is the HERS study, and this is overall death. And you can see that there was actually a higher death rate in the estrogen progestin group than in the placebo group. And similarly, if you look at coronary heart disease incidence from that trial, that was also higher early on in the group that got the hormones. And so there was no suggestion of any benefit and suggestion of some harm. Then there was this huge trial called the Women's Health Initiative that included in this cardiovascular portion, included about 16,000 women, and they saw an increased risk of heart attacks and other cardiovascular events with a hazard ratio of 1.29. That's like a relative risk. And so 29% more women getting these heart events in the women who were randomized to the hormone replacement therapy. There was an increased risk of stroke, 41% increased risk of stroke, and pulmonary embolism. And here are some other events, but I'm not going to go through all these. So basically, you saw from the Women's Health Initiative and HERS that really a randomized controlled trial is what you need to be able to really tell what the answer is. That's going to eliminate a lot of sources of bias, and it's regarded as the gold standard of evidence. There are too many problems with observational studies. You have confounders. You have biases caused by the fact that doctors know whether women are on the hormones or not, taking hormone replacement therapy. Now, some people say that the problem was that the women in the Women's Health Initiative they didn't start getting the hormones until long after menopause, and they claimed that if they had started immediately after menopause, it would have been a different story, and that may be the case. I don't know if there's any strong evidence to support that, but that's the speculation anyway. So I just want to conclude then, observational studies are often the first step to new discoveries. The common observational studies are cross-sectional case control and cohort studies. They can be very useful. All observational studies are subject to biases and confounding. Self-controlled designs, I think, are good designs that are less subject to such biases, because you're using each person as their own control, basically. And I have reached the end, so I will stop there. Thank you.